Hey everyone, last time we talked about ATP, right? The adenine with the ribose and the 3-phosphate groups connected to the 5' carbon on the ribose, all of that making ATP, our fabulous energy molecule of the cell made in the mighty mitochondria. The enzymes that are embedded on all these wonderful folded membranes that go across in the internal compartment of the mitochondria. So our whole point here for chapter 9, learning about cell respiration, is how we make ATP. So in our last lecture, we learned about ATP and a process called phosphorylation. In phosphorylation, a phosphate is given to another molecule, which basically destabilizes it or energizes it so that it can do work. All right. Well, the idea is that ATP has its 3-phosphate groups and is able to transfer its last phosphate to other molecules to energize them. But sometimes we actually have to make ATP at a different level. It's actually kind of an inefficient level. This is called substrate level phosphorylation, where an enzyme, like a kinase enzyme, grabs onto some molecule who actually has a P available, grabs onto ADP, adenosine diphosphate, and gives that P from whatever molecule it is to the ADP, which makes ATP. Now that's one simple, inefficient way to make some quick ATP when you're in a bind and you really need it. We really don't want to make ATP like this all the time if we can help it. There's a better way, and the better way is in the mitochondria, using that enzyme called ATP synthase, which is embedded in the mitochondrial membranes. But there is other ways. So here's another way, plus a great review of phosphorylation. Remember, giving a phosphate group away can destabilize a molecule and energize it, make it change shape so that it's ready to do work. And so we need to make ATP so we're always able to have some on hand and do this. Remember, we can't store ATP. It's a very unstable molecule. So what's the point? The point is to make ATP. So our first part today, we're going to look at harvesting energy. I like to call it harvesting energy before we actually get into the full-blown process of making ATP down in the mitochondria. Let's look at how we harvest energy first of all, how we basically get ready to make ATP. So remember, all the foods that you eat, including the carbs, fats, and the proteins, they all contain potential chemical energy. So we eat those foods so that we can have that potential chemical energy. We digest them into their smaller particles, and then those smaller particles can become our raw materials for making ATP. But all of these processes are going to be a controlled release of energy, and they're going to be a step-by-step series of enzyme-controlled reactions. One little step at a time, remember. We've got a lot of equations here. So let's use glucose as our model molecule to talk about making ATP. We can do it with any of those foods we eat, but glucose is our model that we talk about, and it's the fastest and most efficient way for your body to make some quick ATP when you really need it. Ooh, some equations. So in respiration, it's a catabolic reaction. We're actually going to catabolize or break down glucose to make ATP. That's the whole idea of this. Taking glucose, breaking it down, and then making ATP. So here's our equation. We're going to take glucose, but it will also require oxygen in order to do this. This is why you actually have to breathe, and breathing is one of the most important things for life. Besides water, I think maybe breathing comes first. So glucose plus oxygen, and then from that we'll get energy, but it'll be in the form of ATP. We'll also get water as a byproduct, but that's okay because we're mostly made out of water anyway, so we don't really notice that much as a waste. We can get rid of excess water, no big deal. And then carbon dioxide, which can be a big worry, so we have to have our special system just for getting rid of carbon dioxide. That's the respiratory system. So there's the equations with C6H12O6, and then it's a balanced equation, so it actually takes 6 molecules of oxygen, or O2, will give us ATP in the end. If we really do a good job, it's between like 34 and 38 ATP that we will make per molecule of glucose. We'll make 6 molecules of water, 6 molecules of carbon dioxide, and there will be some heat generated from this, which is kind of nice because I like to stay warm. So this is our equation for cell respiration. You need to memorize this equation and thoroughly understand it and realize how this pertains to making this important molecule ATP, one of the fundamental processes of our lives. So it's kind of like a combustion reaction, really. But in a real combustion reaction, you add fuel and oxygen, and you get a lot of heat, fire. Because we're adding, our fuel is glucose, we're adding oxygen. We don't want to like have fire or anything in our cells, so we're going to use many small enzyme-controlled steps to do this so that we don't blow up. That would not be good. So small little steps at a time, remember that. So how do we harvest energy from our fuels? Well, we have to digest molecules that are large into smaller ones. As we break the bonds, basically, we're going to gather or move electrons from one molecule to another. As electrons move, they carry energy. It's the basic idea of electricity. Electricity is basically electrons moving. So same thing in our body. If we move electrons from one molecule to another, they will actually carry energy with them. It's the energy stored in the bonds. All right, so if we remove an electron from one molecule, we say that that molecule becomes oxidized. Whenever you remove an electron from a molecule, it becomes oxidized. Whoever gains the electron becomes reduced. And that's easy to remember because an electron has a negative charge. So I just think of it, if I get a negative, then I'm reduced. So these are called oxidation reduction reactions, and that's basically what's happening during cell respiration, is series of oxidation reduction reactions where we move electrons from one molecule to the next in a series of small steps, one right after the other. But how do we move electrons in biology? In electricity, they move through wires and such. We get them from batteries and things. But what about in biology? How do we move electrons in living systems? Well, electrons actually just can't move by themselves. They actually have to move part of something else. And so, well, who else has a readily available electron? One only. Well, that would be hydrogen. So when electrons move in our bodies, when electrons move in our cells and move from molecule to molecule, they actually move as part of a hydrogen atom. Remember, a hydrogen atom only contains one proton, one plus, and one electron, or one negative. So literally, I kind of use these words interchangeably. When I say hydrogens are moving, well, the electron is moving with it. So I kind of mean the same thing, that electrons are moving. So electrons must move with their hydrogen. You move a hydrogen, you move an electron. You actually move a proton too, if you think about it. So look at our equation, glucose plus oxygen. That's an oxidation reduction here going from glucose to CO2 on the other side. We're taking away an H. And remember, H's carry electrons. So I take away electrons, or H's, from this glucose. This glucose becomes oxidized. Well, let's see, who gets it? Over here, this O2 gets it. The O2 now becomes water. There it is, the H's. Those H's contain electrons, so it was the water that got reduced. So the carbon dioxide gets oxidized because we take it away from glucose, and the water gets reduced because we added an electron or a negative to the oxygen. And so we have what's called an oxidation reduction reaction going on here, and we're making ATP. All right, so really the main idea here, going to the next one, that these redox, they're called redox reactions for short, they occur in respiration. Our idea is that we keep breaking down organic molecules. So think about glucose. Glucose has, well, it has different bonds. It has 6 carbons in it. We're basically breaking those bonds. Every time we break bonds on these molecules from our fuel that we eat, we're taking off hydrogens. I like to call it harvesting hydrogens because hydrogens carry those electrons that we're going to get energy from. We're stripping off the electrons by removing hydrogens, oxidizing our fuel, literally. And how is this also happening? Well, it turns out oxygen is going to be a big part of this. We're using oxygen because it's very electronegative. It's very readily available, 20% in the atmosphere. We can breathe it in, no problem, readily available there. And it can be available in our mitochondria to draw those electrons towards it. Remember, electronegativity means that you have an affinity or a pull for electrons. So while oxygen is not the most electronegative, fluorine is on the periodic table, but oxygen is really close to fluorine. So it's the next best thing that we can use in a living thing. So oxygen plays a big role in cell respiration as we tear away the hydrogens off of the food we eat. We're going to use glucose as our model. We're going to draw those electrons from those hydrogens towards the oxygen at the end of it all. So the oxygen is a big attractor for all of this. Whew. So we're going to, there it is. We talked about it last chapter. We're going to couple reactions together. Reactions, some of them will release energy, some will need energy. We're going to couple them together. Whew, to synthesize ATP, I get all worked up and excited about this. So this is all review. There's that T chart. So, we're adding more to it. Oxidation, oxidation, I can't even say it. Adding O, but in this case in cell respiration, what we're most concerned about is when we remove H's or when we remove electrons from something, we oxidize it. This actually releases energy or is exergonic. That's a review term that we've already had. Reduction reactions, remove O, but I'm not too worried about that. It's this, adding hydrogens is another way to reduce something, which remember, if you add a hydrogen, then you're adding electrons. This actually stores energy, so it's endergonic. Wow. So moving electrons in respiration. This is a big slide here. We need something to carry these electrons around. Actually, we're carrying the hydrogens around because electrons don't move by themselves. So we're going to use electron carriers or hydrogen carriers, basically. And these hydrogen carriers are different molecules. Here's 2 of the most important ones. These are actually B vitamins, which is why you really feel fatigued and tired when you're low on B vitamins. because then your making of ATP isn't going to be as efficient. You're not going to have enough carriers of hydrogens and electrons. So NAD, who is a B vitamin derivative, niacin, NAD grabs H's. So we're taking glucose, we're tearing the H's off of it, and NAD grabs them and takes them to the mitochondria. FAD is another one, actually can grab 2 hydrogens, so 2 electrons, and take them to the mitochondria. Yep, we need to grab those H's because they carry those precious electrons. So these molecules, NAD and FAD, get reduced because we add an H or an electron to them. And I love this picture here. It's a piggy bank. And when you add electrons to the piggy bank, you have like money in the bank to do work later on, down in the mitochondria where we're going to make ATP. So here's a picture of NAD, what it normally looks like. Don't memorize its form or anything, but you can see here how it can grab this H. Where's it going to put it? It's going to put it right up here. There it is. Grabs an H, so now it's reduced. And then it's going to take it to the mitochondria. It's a carrier. It's a shuttle. It drops it off and then comes back for more. Make sure you go take your B vitamins or eat foods rich in B vitamins. Then you'll be so much better at this, at making ATP. So for our whole idea here is we're going to go through these different metabolic stages to grab hydrogens off of glucose, because we're going to use those hydrogens, we're going to use the electrons in the very end to make ATP. So here's our steps that we'll be looking into in our next set of notes. One of the steps, the first one, occurs in the cytoplasm of the cell. That's where we start tearing apart glucose and start grabbing some hydrogens from it. Then that process moves into the mitochondria, and we have something called the Krebs cycle, which is another kind of complicated cycle where basically all these are is just continuing to break down glucose, keep tearing those H's off of it because those H's have those electrons. And in the end, we're going to take it to this last step. Here in the picture, we have our electron or hydrogen carriers taking to the last step, dropping them off for electron transport. For oxidative phosphorylation, there's that word, where that means that we're going to use oxygen to attract those electrons, because it's electronegative, oxidative, and then we're going to phosphorylate, ADP, to make ATP. So oxidative phosphorylation. I love it. It's so exciting. Whew. Again, what's the point? It's to make ATP. And in one of our last notes, how do we do that? We discussed this very briefly. This is that enzyme whose job, remember enzymes have very specific jobs. Its job is to make ATP. It's our smallest turbine in the world, and inside our cells, an enzyme that actually spins. What makes it spin so it can do work? It's actually hydrogens. This is where the hydrogens are going to go. Remember I said we're continually tearing hydrogens off of glucose, the food that we eat, tear those hydrogens off. We're going to carry them down to the mitochondria, and we're going to gather all those hydrogens in one spot and let them flow through synthase so it spins, so that we can have the P's put back onto ADP, that's that phosphorylation, and then make ATP. About 38 molecules of ATP per glucose that we tear down. Whew. Well, the hydrogens can't just get here by themselves. They need those carriers. They're also going to have the electrons that we gather are going to play a role as well to help pump the hydrogens up here to the spot that they need to get so they can flow down ATP synthase. And also in the end, after the hydrogens flow down, we're going to have to get rid of all these wastes. We're going to have excess protons, hydrogens. We're going to have excess electrons. Got to get rid of those so they can recombine and become a hydrogen atom again. And then we can join that with the oxygen waiting at the end and make water. So we can easily get rid of the waste that we generate from this. But isn't it crazy just to think that the food that you eat, really all you want from the food that you eat for making ATP is the hydrogens and the electrons to take them down into the mitochondria to make ATP synthase work. It's awesome. Wow. Oh, gotta wait until the sequel. Got the energy? Make sure you ask questions if you have any at this point. All right, so in our next lesson, we'll be talking exactly how each of these steps occur. Let me go back to that slide right here. How do each of these steps occur in order for us to get to the point where we can make ATP synthase do its magic? So that will be our next thing. If you have any questions, make sure you ask. And remember, those hydrogens and those electrons, that's where the energy is at in your cells.